this video, I'm going to describe why it is that meiosis contributes to genetic variation. And the key is that the four cells that are the result of meiosis, the four haploid cells, can all be genetically different. And because they're genetically different, the zygotes that are formed from these gametes will also be different, and that's genetic diversity. Now, there are two reasons why the cells that result from meiosis can be different. One is because of chiasma and crossing over, and the second is from independent assortment, which happens at anaphase 1, where one homolog goes to one pole and the other homolog of the pair goes to the other pole. So let's take a look first at crossing over. Now, I'm going to show just one pair of homologs. I'm going to use a straight line for this particular pair of homologs. One homolog is going to be a solid line, and the other homolog is going to be dotted so that we can see what happens when they cross over. Okay, so here we go. There's our first homolog, and those are the two sister chromatids held by a centromere. There's the other homolog, also with two sister chromatids held together by a centromere, and the reason it's dotted is just to distinguish between the two homologs in this pair. All right, so now in prophase 1, they have formed a tetrad, and they can form chiasma if arms of homologs, sister chromatids of the homologs, cross over each other. Okay? And so this area here where the X is, that's the chiasma. And at these chiasma points, the DNA backbone can break and repair so that now we have a leg that used to be over here is now attached to this homolog. And on the other hand, this leg, which used to be over here, is attached to the other homolog. So we've got an exchange of DNA between the homologs. That's the result of crossing over. Now, if we fast forward and go through all of the stages of meiosis and end up in telophase, the way that the four cells look now at telophase is, I'm going to show the four different cells as a result of meiosis. One of them is going to be this guy. The other is down here is going to be this guy. And then we're going to have one that looks like this from here. And then, so those are our four different results at telophase 2. This guy went here. This guy went here. This guy went here. And this guy went down here. So that's telophase 2. Let me get rid of some of this stuff. Now, remember, these are now four haploid cells. I'm not showing any other chromosomes. I'm just working with one chromosome now. So you have to imagine that there are more chromosomes in these. But remember, they're haploid cells. They're going to become gametes, either sperm or eggs. And notice they're all different. This one's all solid line. This has solid through the centromere and then dotted. This has dotted through the centromere and then solid. And this one's all dotted. All four are different, so that's going to contribute to genetic variation. Okay? Now, the second way that you can have genetic variation as a result of meiosis is independent assortment at anaphase 1. So let's take a look at that. This time, I'm going to use a straight line for one homologous pair and a wavy line for the other homologous pair, like I did in the previous video, just where I went through the mechanics of meiosis. And I'm also going to use dotted and solid again to show the two different homologs of the same pair. All right, so here we go. This is showing now a chromosome that's at metaphase 1, or a cell that's at metaphase 1. So we've got, again, our solid. So that's our straight set, and we've got the two homologs of that pair. I'm not showing crossing over or any of that like I just did previously. I'm showing it without any crossing over. And here's our other homologous pair, a solid wavy one, and a dotted wavy one. So one homologous pair, another homologous pair. Now here's the deal. At anaphase 1, remember, the homologs separate. So one of these homologs will go to this pole, and the other homolog will go to the other pole. Now, interestingly enough, which one goes where is never for sure. It's random. So this homolog might go this way, or this homolog might go this way. Now, whichever way it goes doesn't have any impact on which way these guys go. So, here's what I mean. Let's say that for in anaphase 1, remember, these guys split. And so let's say that the dotted one went over here, and the solid one went over here. And then these guys split, and let's just say they split the way they're set up here. So the wavy dotted one goes over here, and the solid wavy one goes over here. Okay, so this is one result of anaphase 1. But it could have just as easily been the other way around, where, for instance, the straight dotted one went to this pole and the solid dotted one went to this pole, but maybe the solid wavy one went this direction and the dotted wavy one went over here. That's a different result. How's it going to be different? Because in anaphase 2, we would have over here, remember we're going to have 4 cells at the end, we would have a solid and a dotted wavy over here, and over here we would have a dotted straight and a solid wavy. So that's one way this could have sorted out. But if we had taken a look at anaphase 2 from the previous example, where both solids went together and both dotteds went together, then the gametes would have looked different. The gametes would have looked like this, where the solids are both together and the dotteds are both together. And those are two different types of gametes, all because of the way the chromosome pairs separated at anaphase 1.